Today we will discuss about this antibody dependent cellular cytotoxicity effect on NK92 cytotoxicity. In the previous video we have checked the NK92 cytotoxicity effect on different target cells without ADCC. So in that case we have not seen any enhanced NK92 cytotoxicity in the genetically modified cells compared to the wild type cells. So the researchers wanted to see whether there is enhanced cytotoxicity when this ADCC is been triggered. So in order to do that, they started with a sample experiment where they identified the amount of drugs or antibody that they want to use for their studies. So they checked with this HER positive cells, that is BT474, co-cultured with this NK92 cells along with this antibodies that are shown here. This trastuzumab, which is affinity towards this HER positive cells, and this rituximab, which is affinity towards CD20 cells. And they did it at different concentrations of these antibodies. And what they're able to see is that the NK92 with this trastuzumab has enhanced cellular cytotoxicity at 10 microgram per ml, where it saturates. So they decided to do their studies on HER positive and HER negative cells with this antibodies, which is trastuzumab antibodies. So when they did the studies on the HER positive cells, they are able to see enhanced NK92 cytotoxicity in the genetically modified NK92 cells. That is for both NK92, which is transduced with CD16 cDNA, or NK92 which is transduced with CD16 CAR, which has the intercellular domains of the 41BB. And we can see that the although both of them shows a enhanced cytotoxicity compared to the wild type NK92 cells, but this NK92 41BB has slightly more enhanced cytotoxicity compared to the NK92 that are transduced with CD16 cDNA. And similar results we can see for SKB3, SCOV3, MCF. When they check it for HER negative cells, they do not see any enhanced cellular cytotoxicity that can be seen in this last figure of the panel B. And thereafter, once they got the results for this HER positive cells, got the positive results from this HER positive cells co-culture, they did the similar studies on CD20 positive cells, that is Raji cells and Dodi cells, in the presence of this rituximab. And here also they are able to see enhanced cellular cytotoxicity when this genetically modified NK92 cells are used, that is NK92 cells transduced with CD16-CAR that has this 41BB intercellular domain, or NK92 cells that are transduced with the CD16 cDNA. So we can see that when only rituximab is used, there is no cell death, as it does not trigger any cell death. And NK92 cells alone cause cell death to a level around 50% at 16:1, but the genetically modified NK92 cells shows enhanced cellular cytotoxicity both for the Raji cells, and similar results they have seen for the Dodi cells. In this case also they can see that the NK92 cells that are transduced with the CD16-CAR that has the intercellular domains shows slightly enhanced cytotoxicity compared to the other genetically modified NK92 cells. So this indicates that the co-stimulatory domains plays a role towards its contributions to enhance the cytotoxicity effect of these NK92 cells. So this is what they finally find out, that without this ADCC effect, the NK92 cells has a cytotoxicity levels which is comparable to the background NK92 cells. That is genetically modified NK92 cells, the cytotoxicity effect which is comparable to the wild type NK92 cells. But with this ADCC effect, they can increase the cytotoxicity effect of NK92 cells against the target cells. This is what they conclude from the HER positive cells and the HER2 positive cells and the CD20 positive cells. So I have said like HER2 as HER in the starting of these videos. So it is actually HER2 positive cells. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.